Hello, everyone. My group and I are presenting the antibody testing method for COVID-19. My name is Bernal.
My name is Maritza Flores.
I'm Wendy Maldonado.
And my name is Jasmine Sanders.
A little background on COVID-19. Discovered on December 2019 in Wuhan, China. Believe that the virus emerged from the wet markets due to lack of hygiene and feces surrounding the animals and vegetables. Cases grew very rapidly, and the first case in the US was on January 20th. And by March 11th, the World Health Organization declared COVID-19 a pandemic. Some of the symptoms that have been recorded after the 2 to 14-day exposure are fever, cough, shortness of breath, loss of taste or smell, sore throat, nausea, vomiting, or diarrhea. Some of the symptoms that require medical attention are trouble breathing, pressure in the chest, inability to stay awake, or bluish lips or face, which signify a lack of oxygen in the body. The antibodies used for the testing are the immune globulin M and immune globulin G. The IgG has two heavy chains that are linked to a light chain by a disulfide bond. This is what gives it its Y shape. Each N has an antigen binding site and it has a molecular weight of about 150 kilodaltons. This antibody has been used as a diagnostic tool for previous viruses such as mumps, chickenpox, hepatitis B, and it's now being checked for immunity against COVID-19. IgM is known to be the largest antibody, and it has a molecular weight of 990 kilodaltons. The structure is made of light and heavy chains, and each chain has a certain amount of amino acids. Research has been done to show that this antibody has a great enhance in antibody response, especially for large cases of antigens. The antigens that are present for these antibodies are the S1 glycoproteins and the S2 glycoproteins. The S1 is located in the protein range of the amino acids of 1 through 674. For S2 glycoprotein, it is located in the amino range of 685 to 1,211. This is the antigen sequence. Due to the nature of this virus, researchers were working endlessly to have a better understanding for it. And on January 12th, China published their first sequence. It can be found on the NCBI with the session number NC045512.2. And up to this day, there are 9,070 nucleotide sequences and 104,484 protein sequences. The structure for the protein S1 is attached to the virus cell membrane by interacting with the host receptor. And for the S2, it mediates the fusion of the virin and the cellular molecular acting as a class 1 viral fusion protein. And for this model, it has been shown that there are 3 conformational states: the pre-fusion native state, the pre-hairpin intermediate state, and the post-fusion hairpin state. The epitope that is present for this antigen is the CR3022, target of highly conserved epitope. The structural modeling further demonstrates that the binding epitope can only be accessed by the CR3022 with at least 2 binding receptors on the trimeric S protein that has an up conformational and a slight rotation. The antigens that are present is the S1 and the S2 glycoproteins, and these antigens act as a molecular key to bind to specific ACE2 cell surface receptors. When S1 binds to the cell surface receptor, structural changes occur, and the binding site of the antigens evoke a strong neutralizing antibody response, which is what has made the development of diagnostic tests and vaccines to be under production.
So we want to know how these antibodies are produced for these testing kits. And the first place that these antibodies are in is the blood and other bodily fluids. The IgM is the first antibody that appears in the body after the first pathogen has entered. IgG is known to be the most common antibody because it protects against both bacterial viruses and infections, and it could last in the body for months to even years. Next, we want to know what the key components involved for making the testing strips. And as we see the picture above, the testing strip contains the adhesive card and the sample pad on the left with a conjugation pad following. Next is the nitrocellulose membrane with 3 lines. Those 3 lines are MG and a control line. Finally, we have the absorption pad that collects all the excess flow, and that will be explained later in the presentation. Next, we want to know what is the lateral flow immunoassay. And the lateral flow immunoassay is known to be a quick way to achieve results within minutes by detecting large and small molecules of the virus. It's a binding reagent that's technically coating the protein as an antibody, and it blocks the unoccupied sites. So by incubating the primary antibody, it becomes easier to match the target virus.
Next, we want to know the materials and methods on the rapid test and the ELISA test. So the instruments used on the rapid antibody test is the detection cassette. This gives a positive or negative result. The lancet, which is used to draw blood from the finger. The pipette, which is used to collect the blood and place it into the detection cassette. And the buffer, which is used to dilute the sample. Instruments used in the ELISA is the microwell, which must be coated with the COVID-19 protein, and the microplate reader, which must be read at 450 nanometers. The assay for rapid antibody test functions when once the blood sample and 3 drops of buffer is added into the detection cassette, the sample will then enter the testing well and the COVID-19 antigen complex will bind to the IgM and the IgG antibody. This creates the red blood cells to migrate and yield a color change on the detection line of the C-control IgM or IgG. For the ELISA plate is covered with the antigen found on SCOP2. These antigens are proteins found in the virus. Once the blood is obtained from the patient, the red blood cells are removed and only the clear serum is kept, which contains the antibody. The serum samples are added to the wells of the plate. If the specific antibody binds to the antigen found on the ELISA plate, wells are then washed to remove non-specific antibodies. Secondary antibodies is then added and washed. The substrate binds to the secondary antibodies, a reaction occurs and produces a visible color. So how fast is the testing? For the rapid antibody test, it's pretty fast, which is around 5 to 30 minutes, and it tells you the presence or absence in a qualitative form of antibodies against the virus present in the patient. For the ELISA test, it can take up to 2 to 5 hours and indicates a presence or absence in a quantitative form of antibodies against the virus. So how is the positive result determined? For the ELISA, when the substrate is added to the plasma, it would react with the enzyme and create a color change, indicating it has the antibodies that will bond with the antigen found on SARS-CoV-2. This results a positive test result. The IgM and the IgG rapid test, red blood cells will migrate across the device slowly. Positive results creates a color on the C, IgG, and IgM detection line, or color on the C detection line and IgM. Finally, it can also create a color change on the C control line and the IgG detection line. How do we interpret these testing results? So it is important to know the antibody tests do not test for current infection. For the positive result, it indicates the presence of antibodies which comes from the infection of SARS-CoV-2 that causes COVID-19. IgM antibodies have been detected, color on the control line C and IgM detection line. For the control line C and the detection line IgG lines are both colored if the IgG antibody is detected. If the IgG and the IgM antibody is detected, control line C, detection line IgM and IgG are all colored. For a negative result, this indicates no infection or antibodies have not yet been developed. There exists a possibility infection can occur or has occurred. No quality control line C is colored, IgG and IgM lines are not colored. For an invalid, this indicates that the assay is not working properly, control line C is not colored, even if the IgG and the IgM is colored. So how accurate is this test? The antibodies test 1 week after first symptom detects 30% of the people who had COVID-19. Accuracy increases in week 2, detecting 70%. At week 3, more than 90% are detected. After week 3, tests gave false positive results. For example, if 942 people will test negative for COVID-19, of these 4 people, or 0.4%, will actually have the COVID-19. This will give a false negative result. Sensitivity or true positive rate measures how often a test correctly generates a positive result for people who have the condition that's being tested for. A test containing a high sensitivity will not generate many false negative results. Specificity or true negative rate measures a test's ability to correctly generate a negative result. This test will correctly rule out almost everyone who doesn't have the disease and won't generate any false positive.
So what reagents are in the sample well or sample pad and reasons for their use? The CDC 2019 ENCOVE Real-Time RT-PCR diagnostic panel contains 4 reagents. There's 3 primer probe mixes for 2019 ENCOVE N1, 2019 ENCOVE N2, and RP. The 4th one is CoVPC, which is a non-infectious positive control material that yields a positive result in each assay, including the panel. As mentioned before, the reagents that are used on the testing line are IgM and IgG, with IgG taking up approximately 80% of the total antibody count. So what is visualization method and what is the principle for it, and how are the antibodies attached to the particles? Cassette-based systems is a form of visualization method in which can be read at the point of care through a change in color and an indicator region visible to the naked eye. Cassette systems use the principle of lateral flow immunoassay or immunochromatography. Antibodies from the serum bind to antigens in the test strip and are wicked laterally along the length of the test strip. In the indicator regions of the test kit, anti-human antibodies, which are immobilized in the test strip, will bind to the antigen antibody complex and hold them in the indicator region. Are colloidal carbon or latex particles used? So the colloidal gold or other colorant accumulates in the indicator region, leading to a visible change in color along a narrow band of the wicking substrate. The control line is an antiviral spike antibody, uncaptured viral protein coated colloidal cold or colored latex, wicks past the antibody class specific test lines and pitches up on the control line. All tests should contain a control line to indicate that the test has functioned properly, specifically that the viral protein coated colloidal gold or colored latex has wicked along the complete length of the nitrocellulose or paper substrate and over the class specific anti-human immunoglobulin lines. So the cassette-based systems rely on a color change that is visible to the human eye rather than an instrument. This is more appropriate for the use of care as it can give immediate results to healthcare providers and patients. The typical lateral flow immunoassay cassette, shown at the right, has a control band marked C and 2 antibody indicators. The M band indicates the presence of IgM and the G band indicates the presence of IgG. For a test to be considered valid, the control band must be present. Therefore, the test cassette on the left would be considered a negative test result, whereas the cassette on the right would be an invalid test result. Results and discussion. So it is important to understand the cross-reactivity to other viruses of the test being used so as to avoid misinterpretation of a false positive result that may be detecting an antigen from a similar virus. Molecular testing is imperfect as well, with up to 30% of false negative rates in some series. Therefore, when looking at the sensitivity and specificity of antibody testing, one must be academic enough to recognize the inherent weaknesses of using such a test to evaluate another test. It will be hard to know whether a positive antibody test in a patient with a negative molecular test means the former test is a false positive or the latter was a false negative. These are the current cases in California per the California Department of Public Health. There's almost 300,000 total cases. Most are in the age range of 18 to 49 and majority being male. Currently, there are about 6,500 fatalities.
In order to wrap this up, the advantages of this test is the accessibility for people to go ahead and take the test. But what's important to understand is who has the virus and who might transmit it to others, and who's had the virus and might be immune to it, as well as who might be potential help to others for future treatments, such as disease prevalence, vaccine development, or plasma therapy. And as there are advantages, there are also disadvantages, and the test quality would be one of the disadvantages, which means that there's some that don't perform as best as they can, and it's still being evaluated by the FDA. And sometimes there's no signs of the antibodies in the patient, so they could be exposed to COVID-19, but they don't develop any antibodies. As said before, the false positives and the uncertain immunity, which we don't know if you could catch COVID-19 again after you have already been infected. In order to go ahead and improve that, of course, we'd have to go ahead and find a cheaper and better quality for these tests and decrease the false positives, so eliminating any doubt of any other viruses and finding a way to make the tests more sensitive to pick up other signs of undeveloped antibodies. And of course, keeping up with the disease prevalence.